biomolecules form droplets. We call these biomolecular condensates. We've been seeing about phase separation, so I won't go into what this process is. And here I've just shown some examples from cells and in vitro the formation of these droplets. So these are droplets in the bulk solution, but these droplets can also form on surfaces. Inside cells you have various surfaces such as polymers, membrane, and so on. So again, I've just shown a few examples from the recent literature. So you have droplets forming on various surfaces, both inside cells and in vitro. And one thing to remember is that these droplets can also form below the saturation concentration required for bulk phase separation, right? So when you are at concentrations even lower than CSAT, you can have these droplets forming on the surfaces. So the physical principles behind this condensation on surfaces, it's not completely clear, so that's what we want to understand, but also we are motivated by a very specific biological system, so I'll come to that. So I mean, like I had mentioned, tau proteins and microtubules. Just a quick background on what these are. So I mean, microtubules are these really long stiff polymers that provide structural support to the cell, and I mean, they are composed of these protofilaments, but yeah, essentially you can think of it as a very stiff polymer. And tau proteins are small proteins which are usually found in the axonal region of neurons, and they perform a host of functions like stabilizing the microtubule, regulating the transport of molecular motors, and some other functions. Tau is also very interesting biologically because it's studied a lot in the context of Alzheimer's disease. So tau can get hypophosphorylated and it forms these aggregates which in downstream it kind of causes Alzheimer's. So even like biologically it's a very interesting system. So I mean, the experimental setup that our collaborators have is that it's an in vitro setup. So they take a microtubule, stick it on a glass cover slip, and introduce fluorescently labeled tau proteins. And okay, so this is what it would look like. So you see that this condensation begins and then it grows. It spreads on the microtubule, and then it stops. It doesn't completely cover the microtubule surface. So okay, yeah. I mean, like I said, once it stops growing, this is what it would look like under a microscope. So I mean, already you can see there are these really bright regions, and then there are these. Well, it's not showing that great on the slides here, but there are these dim regions as well, right? So already there's the question that why is it not uniformly covering the entire surface? Why is it doing, this kind of why is it showing this kind of a behavior? Also, if you look at the intensity distribution along the microtubules and you plot it, you actually see that there are two peaks, which visually also you could have guessed. So it means that there is preference for two states, right? There are two distinct states that these molecules are going to. And you can do back of the envelope calculations to actually show that the regions in this, like in this dim regions, it is actually just single layer binding, just adsorption, simple molecule binding to the surface. And in this thick, brighter regions, I mean, I call it thick because you can actually show that it's a multi-layered thing. So it's not just a protein binding to the surface. So first layer forms, and then another layer of protein will bind to this layer of protein, and so on. And you can estimate that something like 3 to 5 layers are forming here, right? Then there is another feature that as we increase the concentration, the fraction of microtubule covered by these thick regions, which I call surface condensates, like interchangeably, they increase, and we'd like to quantitatively understand this data from the experiment. And another interesting feature is that if you take a microtubule and once these islands form, you can wash away the tau protein and these islands disappear, and they can again introduce the tau proteins in the same setup and they'll again form, and you can do this in repeated cycles. what you see is that these islands always form at the same location, so their positions are not random. So with this specific system, how do we understand these results? How do we model them? So that's what we want to do. So we'll just use a simple lattice gas model. I have a 3D lattice, and each site can either be occupied by a protein or not. It's a very coarse-grained system. So yeah, you denote it by 1 if a particle is present. By particle here I mean a single molecule of a protein, and 0 if it's absent. And then you can write a Hamiltonian, which, well, you have interactions for like bulk interactions on the surface, which I'm showing here in red, the affinity of a single molecule to come bind to the surface, and also I allow for the number of particles to change, and the overall concentration is kept fixed by imposing a chemical potential, right? So just a grand canonical Monte Carlo simulation. And what first, of course, lattice models we use for like simple phase separation we can do, so you get those same, the now familiar binodal you get, and you can get these phase diagrams, but this is all in bulk at higher concentrations. And now when we introduce a surface similar, kind of respecting this geometry of a microtubule, then what do we get? So what we see is that so as you increase the concentrations, you get this surface condensation. Note that you don't have any droplets in the bulk. It's not stable there, right? It's only near the surface that you can form these multi-layered structures. So it's the same thing that I'm showing again. Like, this is the top view, and so at lower concentration, I haven't indicated the concentrations here. Numbers don't matter. It's qualitative for now. At low concentrations, you have this kind of a single layer sparse binding. This is just like a what do you call adsorption. There's nothing special here. But as you increase the concentration, you actually get like multiple layers, something that you can call like a condensate or something that's thick. To give some physical intuition behind this, I'll just quickly talk about some of the physics that motivated us with this picture. So again, this is the familiar binodal that we have. So you have concentration on the x-axis, temperature y-axis. And inside this region, you have bulk phase separation. We are not in that regime. We are at concentrations lower than this. So if I look along the red line, we are at lower concentrations than this one, right? And thermodynamics, using thermodynamics, even in the 70s or 80s, people have shown that there exists this first order transition line, which actually separates this adsorb state and a condensed state. What I mean by adsorb state is the single layer sparse binding, and the condensed state is actually a multi-layered, multi-layered wet thing on the surface. And this is a first order transition. And our hypothesis or our idea was that the fact that we see these two states with a thin layer and a thick layer, then maybe the system is in the vicinity of this first order transition line. So yeah, this is just a cartoon representation of what I was mentioning. So as you go along the red line, initially it's a thin layer, and it goes to this thick layer, and then of course if you go to much higher concentrations, you have droplets forming in the bulk as well. So the key takeaway with that is that the protein-protein interaction, it's not strong enough, or the concentration is not high enough that you can form droplets in the bulk, but in the presence of a surface, locally it can get stabilized, and you can form these multi-layer structures near the surface. So what we saw, we do see these thin and thick states, but still what we saw was that it covers the entire microtubule. So then the question is, why don't we see coexistence like in the experiments? So we thought that, okay, maybe the microtubule surface is not homogeneous, and I had a term showing the affinity of the molecules to the surface, and we just, instead of keeping a constant value, we draw it from a normal distribution, right? And then we see that it shows something like this, right? and now you're seeing coexistence of the thin layers, for example, in these regions, and then you see the thick regions, which is very similar to what we see in experiments. And here also, if you do what you call a histogram of the density along the microtubule, you see a bimodal distribution, which is again qualitatively it's like similar to what we see in experiments. So then this hints that the surface has to be heterogeneous. So yeah, before going to that, like another like interesting output from the model is that this persistence of the same patterns or like the same patterns arising for a given microtubule is also something that comes out of the model without actually putting anything special in that sense, right? Like so here I'm just showing a randomly drawn like affinity, and you see that I mean I've just shown 4 representations here, but for individual simulations starting from different initial conditions and all, more or less your patterns are reproduced. So again, you don't need to put something special for this to happen. So now yeah, this is just a quick thing I'll just show to make like quantitative comparisons to the experiments, because what we saw for now was qualitative. We like coarse grain this to a 1D system, because the system has cylindrical like azimuthal symmetry, and you have the thin or the thick states on the microtubules. You can essentially write a 1D description for the system, and you can say that at each state it is either at each site in the 1D lattice it is either in the thick state or the thin adsorb state, right? And yeah, so show it by +1 -1 and write an effective like energy description. Formally it's like a random field Ising model, but then how we interpret it is different, and then you can relate these terms to actual concentrations in the experiment and make quantitative comparisons, and also to the kinetics. So again we show that I mean you see that like a homogeneous surface can kind of capture the equilibrium data, but if we look at the kinetics, then it doesn't quite capture the trend. So we're looking at a chymograph. So in this direction it's time, and this is the position of the microtubule. So in the experiments you see these nice stable islands which form and then they stop growing. But with a homogeneous field, once these islands form, they just keep moving around. So the idea is that because there is no energy penalty for these islands to like move once they've formed, they don't form stable islands. But now if you break that symmetry and you introduce heterogeneity in the surface field, you can give rise to these stable islands, similar to the experiments, right? So now just to like kind of probe the source of this heterogeneity, because microtubule is a homogeneous kind of a surface. It's made up of a single it's made up of a single type of dimer. So what could be the source of this heterogeneity? So so one thing that was a possibility was that post-translational modifications of tubulin are playing a role. So I mean, just to give an idea, it's like so these are phosphate groups that can be attached to the tubulin protein after they are like created, and that can introduce this heterogeneity. So to test this out, like what our collaborators did is they take microtubules from 2 different sources. One has a lot of PTMs, one has very few PTMs, and then they break them apart and mix them and form this kind of a construct which is mixed, and then introduce the tau proteins. So in our model, this would correspond to like the low PTM regions would correspond to turning off the surface heterogeneity in some regions, right? And from the model what we see is that like the prediction is that it doesn't form islands in this region where there are no PTMs, and here they do. And from the experiments what we see is the same thing, which kind of again like validates like kind of the model. So here in the pink region you are seeing the high PTM microtubules, and here you are seeing the low like PTM regions, and the islands are forming in the high PTM regions. Yeah, just yeah. So so the key takeaways. So the first thing is that you can form these surface condensates even below your bulk phase separation, like saturation concentration. And this kind of a framework, because it's a very simple coarse-grained model that we're taking of protein-protein binding, protein-surface binding, you can extend it to other like biological systems. It's a very general framework. We are looking at specifically tau microtubules, but it can be like anything. And like specifically for this system, we say that we find that you need the surface to be heterogeneous, and it can come from post-translational modifications, and well, cells can harness this process to regulate all kinds of like downstream processes. So, yeah, thanks.
So you are modeling this by gas model.
Yeah, lattice gas. Yeah.
Lattice gas.
Yeah.
How are you solving this?
Monte Carlo simulations.
Monte Carlo simulation.
Yeah.
So can you go to that Hamiltonian?
Yes.
So the first term, it is already including the surface term.
No, this is for like everything like not shown in red. So this is for everything above. In the red region, it is these two terms that kick in, the surface terms.
And then you are applying the field only on the surface, not in the bulk.
Yeah, I mean, on the surface there is this field HI, which accounts for affinity like of a single protein to come and bind to the surface.
And then this is you are putting some.
Yeah, chemical potential, because I allow the number of particles to change. I can remove or add particles, but you still want to impose a fixed concentration in the solution. So a chemical potential does that. So grand canonical laws.
When you map what you are saying, when you map to the one dimension.
Yes.
You are also solving a Monte Carlo that one dimension.
Yeah, I mean the homogeneous one you can do analytically, heterogeneous one you can do numerically. You don't need to do simulations, but. Yeah.
So when you tell you introduce heterogeneity in the simulation, what does it mean?
So this HI you see, right, which is like the affinity of a single molecule to bind to the surface. In the homogeneous case, it is same everywhere. In the heterogeneous case, for each site it is drawn from a randomly drawn from a normal distribution.
So the affinity you mean?
Yeah, exactly, the affinity.
So you said this, I don't remember, I think you said the islands always form with a specific
Yes.
location and not random.
Yes.
Why does that happen?
So.
Because when you put on more concentration, anyway, you don't see that effect, right? Because everything comes in.
Sure, at higher concentrations it completely covers. Yes.
So what happens at the low, why there is a specified location?
Right.
That's one. The second, how do you actually implement that in the simulations? Because simulations are also random.
Yes. So the fact that we are saying that the surface needs to be heterogeneous, so there is something that's breaking the symmetry of, there is some underlying potential. But from the random surface, like random potential itself, there are some regions which are like maybe average regions which have a higher affinity and some which have lower affinity.
Simulation let's say that you can essentially identify some islands where
Yeah.
That is what heterogeneous part is doing properly, right?
Yeah.
But in the experiment, why do you see?
So in the experiment, you can't directly probe that landscape. So yeah, I mean we think that like something like this can be indirectly maybe used to make a statement about like even the fact that post-translational modifications are playing a role, like use this to make an indirect like statement like that. Because directly you can't probe and say that this area has this attribute.
Yeah, good question. So you showed two modes of distribution.
Yes.
Right? And you compared with the experiment.
Yes.
Also, right? So the question is next one.
Yes.
The question is, how sensitive is this distribution to the concentration of your protein?
Yeah. So yeah, that's there because if I go to much higher concentrations, I mean everything gets covered.
Yeah. So
And
therefore, is it legitimate to say that there are two phases? Or simply it's a coverage, absorption? I'm just asking a theoretical question.
yeah. Can you? I didn't quite get the question.
I understood from your description that there are two phases that are coexisting.
Yeah, the thick layers and the thin layers.
Maybe they are not phases because it's sensitive to the concentration. So matter of basically how much nutrients, how much proteins you have to cover available sites.
But they have particular structures, like a bimodal.
No, that's why
What he's asking is that is it an availability of the protein that is dictating it? Is it possible that you are forming these thick phases in some areas, and then you do not have enough proteins in the system to form the other thick layers in the other ones? So in other words, if you're increasing the concentration, your availability the availability of proteins is going up. Now you can cover the whole thing. Is that the phenomena, or you have something else?
Right, but even in that case, I mean, like, so even experimentally, that's something that's is seen that as you increase the concentration, the coverage does increase, right? But even at these intermediate concentrations, the fact that you will get a bimodal distribution is not something that is that obvious, and that this is an emergent phenomenon, right? Because whatever surface interactions have put are only here. Whatever is arising, like after the second layer, that's emergent. So the fact that you get a bimodal like distribution, even in intermediate concentrations, it's not.
that wouldn't guarantee that there are two phases.
It's not a
It's not as the measure is. We can talk more. Good job.
Looks like a picture of a
Where
space.
are you going?
I know. It will take us somewhere. So we stop.
Some technical issues, but okay. So,
Can I introduce you before you?
yes, sure.
So Roni, everyone knows Roni. Roni is working with one industry and also so I'll start here and then you'll hear the noise, even if I'm not here.
Hello. I'm audible at the back, right? Yeah. Okay. So the title of my talk is Interaction between Model Bacterial Membrane and Synthetic Ternary Polymer. So my work mostly involves doing all-atom simulations, and I mostly look at gram-negative bacterial membranes and their interaction with something I am terming as ternary polymer. The polymer is ternary because it contains three types of groups. It contains a positively charged group, hydrophobic group, and a negatively charged group. So before I go into the nitty-gritty of the talk, I will first give some introduction to something called antimicrobial resistance, which is a leading cause of hospital deaths. So generally the idea is this, that you have an individual which has a population of bacteria which is responsive to certain drugs, and in this same population there are some bacteria which are resistant to the drug as well. You take the antibiotic which kills most of the bacteria, and if you stop taking the antibiotic without completing the course, what will happen is now this initial starting population will have more amount of resistant bacteria which will grow in high numbers once the drug has been stopped, and now the patient will have too many resistant bacteria and become irresponsive to the drug that was being given. The concern here is that bacteria have something called horizontal gene transfer. So the gene for this resistance can be passed to another non-related bacteria, and that bacteria will also turn resistant. And once this resistance has been acquired in some species, it like rapidly spreads throughout, and then you have huge amount of trouble administering medicine. And this is a picture which is showing that projected number of deaths related to antimicrobial resistance, and you can see that by 2050, there is some exponential increase kind of scenario. So a rather bleak picture, and in this case, the researchers have been trying to find avenues which don't use antibiotics, use other sort of compounds to tackle the bacteria. And one sort of compound which people are looking at is something called antimicrobial peptides. So over the course of evolution, nature has created some proteins which disrupt bacterial action. So these proteins can work in a myriad number of ways. They can disrupt bacterial membranes, they can recruit immune system to specific sites, and other sort of things. But now the problem with these peptides is that once you are working with peptides, you need to synthesize them, you need to purify them, and doing that at scale is slightly tricky. So what people have been looking at is another kind of polymers, which is called antimicrobial polymers, and they are sort of mimetics of these antimicrobial peptides, and they are easy to synthesize. You can also control their chemical properties by tinkering around with what specific monomers you are picking, and these, since they are not biological compounds, so they are also resistant to chemical degradation. So the bioavailability of the drug is higher. So this three molecules which I am showing are something called, the MA stands for methacrylic acid. So these are polymers of methacrylic acid. This is the hydrophobic one. This is the positively charged one, aminoethyl here, and this is the propionic acid, which is the negatively charged one. So these three monomers will make the polymer which I am studying. So here I have color coded them as hydrophobic is green, positive is blue, and negative is red, and this same color code will be carried out throughout. So for our study, we took two different sequences of polymers. One is a block copolymer, where all the charge groups are placed together, and the other one is a random copolymer, where the charges are distributed evenly at random. And for the membrane interaction studies, we focus only on the first one, because the this one sort of has a very trivial sort of thing, where only the hydrophobic group will partition into the membrane. We take both of these sequences and study the properties of the aggregates and so on, but for this talk I will not talk much about the aggregates and instead talk about the membrane interaction. So here I am showing the initial structure of the initial configuration of the system. So here these yellow and blue lines which you see are the bacterial lipids, and this membrane is composed of two different lipids. One is POPE and the other is POPC. POPE carries a positive charge. It's effectively neutral because you have a phosphate and a positive charge. The POPC on the other hand is effectively negatively charged. So what we see is that over the course of a simulation, which is we took two sets of simulations, both of them we ran for near microsecond long. We see that some polymers do partition into the bacterial membrane, and we also see that the hydrophobic groups are the ones which are partitioning, the green color, and the other groups are sort of being pulled in because the hydrophobic groups are being partitioned. So now what we want to quantify is that what are the effects of this sort of polymer partitioning into the membrane, and we mostly look at two different measures. One, the measure on the left, we are looking at the membrane thickness, and the membrane thickness we are measuring along the XY plane, and we see that when we look at the control case where there is no polymer added, the membrane thickness is comparatively less on average when compared to addition of the polymer. We also notice that when the polymer is added, the average membrane thickness goes up, and the difference between the minimum and the maximum thickness also goes up. So and we then look at the organization of these lipids on the membrane. So what we are looking at is that in the black case where the control, and we have the AMP, and the AMP, the antimicrobial polymer, is added only on the upper surface. So the lower surface acts as sort of a control in that case as well. But what we notice is that upon the addition of the polymer, both the upper and lower leaflets show some change in lipid organization when we are looking at POPE to POPE correlation and POPG to POPG correlation. The cross correlation of the lipids are mostly unaffected, but they do change when compared to the case of the control. So together what we can say is that the polymer is sort of changing the membrane properties. Both the change in thickness and membrane organization can lead to some possible disruption of the membrane integrity and possible lysis of the membrane. We also look at something called lipid packing defects. So the definition of the term deep defect here goes something like this. So we have a membrane lipid which has like these two tails, and at the water membrane interface, because of thermal fluctuations, sometimes what will happen is these tails will get exposed to the water surface. So if this exposure which is happening is below the glycerol carbon in the lipid, we call it to be deep. If it's above the glycerol carbon, we call it to be shallow. So the reason to specifically look at these defect sites is that research over the years has sort of highlighted that these lipid packing defects are key to the recognition of these polymers or peptides to the membrane surface. So they mostly detect these defect sites, stabilize them, and thus partition in. So what we observe is that upon the addition of our polymer, the deep defect sites on the upper bilayer, upper leaflet of the bilayer where the polymer sits, goes up by a large extent, whereas the shallow defects are unchanged in the case of both upper and lower leaflets. So here the lower leaflet is sort of like the control where the polymer is absent, right? So what we can say from this is the upon addition of the polymer, the total lipid defect area, so the total defect area of the deep defects goes up. We also see that the number of deep defects goes up, as well as the average area of these deep defects. So these are the distributions of these defects. So we see that the addition of the polymers changes the defect in the upper layer where the polymer sits, and we see that the total defect area, sort of the distribution shifts to the right. So you see more larger deep defects. Continuing on, we also look at the properties of the water at this membrane interface. So the water is also a key participant in recognition of polymers to biological membranes, right? And what we look here is at two different quantities. One is the total residence time of the water, and another is the survival probabilities of the water. So the way we define the interfacial water here is that first we look at the GR of the water molecules when compared to the lipid surface, and we classify water within some 3.5 angstroms of the membrane average membrane height to be interfacial water. And once we have these waters, we can just trace the time, total amount of time each water is spending in some specific layer, and we see that when you have the polymer added, the sort of the residence time distribution has now longer tail. So in the sense that waters are now staying for longer periods. So for these very long-lived waters, we observe that these waters are sort of sandwiched between the polymer and the membrane, and since it's now held in place, it sort of can't escape in this scenario. So but the other ones are mostly being stabilized by charge interactions, since the water has some since the polymer has some positive and negative charges. The charge interaction is holding the waters in place, increasing the residence times. So another quantity which we look at is the survival probability distribution of these waters. So the way we define survival probability is that we have this term where the D here is marking a specific layer, and we are looking at we are tracing some ith water molecule, and we are saying that the value of this Pi will be 0 if the water molecule is not present in this layer. It will be 1 if it is present in the layer, and then basically if the water molecule leaves the layer at any point of time, this term goes to 0 for that water molecule, and we sum it up over all the n water molecules here. So after you do this, you compare it to some reference point. So in this case, we take a reference point as S0, which can be any window in the simulation. So we are taking some specific window, and at that window we take this the initial list of water, and then we can trace this this ST quantity, and after the ST quantity has been derived, we can sort of fit a bi-exponential curve. So the bi-exponential curve here sort of has a fast time scale and a slow time scale. If you compare the time scales here, you will see that upon addition of the polymer, the slow time scale changes a lot. The fast time scales are still comparable. So the fast time scales are the inherent dynamics of the water. The slow time scales are what is being sort of affected by the polymer. So overall, you can also say that the presence of the polymers is changing the properties of the water at the interface as well. So finally, for acknowledgements, I would like to acknowledge Professor Vani Memparala, who is my guide here, and the interfacial water portions of this work was done in collaboration with Professor Anand Debrant at IIT Jodhpur, and I also acknowledge the high performance clusters Nanda Devi and Kamit.
One question I have, so regarding this y exponential.
Oh, this doesn't work. Huh.
So, I mean, here, I mean, you don't have enough steps. Is it like
This one?
Or is it the tail? How you, I mean,
Oh, this one.
How expensive is to get?
So so we have 2 microsecond long simulations. So most of these ones, the extremely long lived ones, are sort of like waters which are sandwiched. So these are like very rare cases. It's not because the sampling is slow. It's just that not too many waters will be sandwiched anyway. So these ones are sort of like just a very few edge cases. So it's configurational in some sense.
And also, how do you choose the bin size?
So so the bin size we chose so that the
Because I'm not seeing error bars.
so here, like, I wanted the distribution to look smooth. If you take too large a bin size, you still see the exponential, but the getting the, like, when I'm doing this calculations where I'm trying to fit the by exponential, then there is too much divergence. So I chose a bin size that the variance in this tau slow and the tau fast sort of goes down, depending on the sliding window thing.
So the plot 2 and 1, how they are connected?
So the plot 1, I'm looking at a single water molecule and I'm seeing how long this water molecule stays in the interfacial layer. The plot 2, I'm seeing that how many water molecules stay in that interfacial layer for, let's say, this amount of time. So if I am saying that the survival time is 20 picosecond, it means that it will be 20 picosecond by which one water molecule will leave from this initial whatever S0 number of water molecules to the other layer. So this is a survival plot. This is just the like mean times or water molecule standing.
But you are extracting AF and AS
From this one. I'm fitting the AF and AS to this plot, not this one.
But this is a theoretical curve. Huh? Where is your
Oh, the data are these triangles at the back.
Yes,
So
Okay.
yeah, it matches quite nicely with the prediction. Yeah.
So, quick question. So, in the real world, right, do you have a comment about whether the membrane is pore is created on the membrane?
So in this one, since we can't, like, the problem in pore generation once is you have to do replica exchange and stuff because otherwise the barrier is too high. We did not do a replica exchange, so this one we don't know whether it can form a pore or not. But judging from the sequence that it has, like, if we look at the sequence here where the charge distribution is random, so to form a pore there will be a very high energy barrier because lots of the charge will have to be first embedded in through this bilayer. The polymer is long enough to bridge, but it will have to be, like, stabilized by interaction with water and ions for it to, like, form a pore here. So I think in most cases this will work as a surfactant kind of a thing where it carpets the membrane and then sort of, like, takes patches off. This very likely is not a pore forming one because most of the pore forming sequences you will see that there is a hydrophobic, like,
so by design you provided pore form.
Yeah, this one by design should not form pores mostly.
Because you have these long-lived states, right? So at least you have few.
Hmm.
Like, how do you actually reason that it is restricted about explanation?
In this one,
Yeah, because that will also reflect in the survey.
okay, so for this ones, the survival probability we are what I do is I take the entire simulation and I am choosing only some 50 picosecond windows over the simulation time frame. So this 40, right? I am taking 50 picoseconds and sliding it around. So the long-lived ones, they will be a very small fraction. So they will, I think, not contribute much. Like they will not distort the data too much.
Yeah, but he's asking why do you take two exponential, not
Exactly.
three,
So-
four, or stretched?
Oh.
There is something else also.
If I can add to Arnav's and
I see.
I think your question was related to Arnav's thing as well. If you do not have a data error bar on the data, so whether single exponential is a good fit to the data, you do not know that. Whether you really need a double exponential,
I see.
I mean, what are the tail contributing to? So that is one thing. Without error bar, I cannot say. I can still have this one with a very small error bar, and I need 3 exponential, and I can have this one with a bigger error bar and a simple straight line. I get that as well.
No, you already have 5 parameters.
Whether these 5 parameters are needed
I-
or even 2 parameters are
So we
enough.
were just following some methodology which has been followed for water dynamics, but for this specific scenario, whether like we can do away with fewer parameters or not, that I have not specifically checked. I was just following the set procedure. I can try fitting them with single exponentials and see.
No, I think going back to Arnav's earlier question, we could fit it with a stretched exponential. That will show a cascade of all these projects. In principle.
Okay.
When you have multiple exponentials, it is equivalent to a stretched exponential.
Yeah, but the first figure he has a nanosecond, right? This 0 to 5. Second figure you are plotting.
We are plotting in picoseconds because I am looking
so
at.
That's why I asked earlier. That first curve and second curve. Your data, where it is reflecting this in this curve. You said no connection.
No, it is connected.
It is connected because the survival probability is related to the residence time, but
Then the second question, the effect, what he is asking, the nanosecond at 5, right? That is, you have just 2 points, right? At 3.6 and 5. Right? The rest of
these
the
ones,
places,
yeah.
I don't see any data, neither for blue nor for the AMP.
No, the blue ones is the control case in which these ones are long-lived because they are sandwiched between a polymer and the sort of. So these ones are stuck.
Available. What is the meaning? I am not getting a survival probability.
Since the survival probability, we are looking at a much smaller time scale compared to this.
So that is only that curve you have. No? So that's the he is asking this by the whole region, the blue and this 0 to 1.5 is just you are blowing up this region, no? In the picosecond. That you can say.
That is usually this is the sample you have taken from 0 to 1.5 more or less, right? To actually construct the plot below.
This one?
Yeah.
No. We take the entire 5 nanosecond duration.
Then that is really the confusion because you have some long lived states.
Yeah, so we take the entire 5 nanosecond window and then I choose 1000 50 picosecond time windows
The
at.
long lived states, right? In some sense.
Because I'm choosing randomly, they may or may not.
This is not a very
But is that the set procedure? Because you said you follow the procedure.
Yeah,
So in the set procedure, do you eliminate the previous works also eliminate these long-lived states?
So in the so those ones I don't exactly know whether they eliminate the long-lived state in that one or not. I know that they have taken sliding windows of this duration, and since those work they did not provide the residence time curve actually. So the data in the paper which I am following is mostly on the first choosing the interfacial bilayer by doing the GR plot, and then once you have the bilayer, you are just looking at once you have the layers defined, you are just looking at survival probabilities in each different layer. So, like, I also would like to say that the entire 5 nanosecond time is not being used for this simulation. Sorry. So there is a smaller, some 100 picosecond long.
No, if you put this 40 picosecond, that's a point or 0.04 nanosecond.
Yeah, that's a.
Right.
Right. So we have a. So to do this survival probability calculations, we have. So this one we are writing data every 20 picoseconds or so. The water survival probability is in that order. So we have lower 4 picosecond time duration data, and that was done for like a few hundreds of picoseconds. So this is from that data. So here we don't exactly have these extremely long-lived waters. This is from a different set. So I think that was causing the confusion. So these are from two different simulations, kind of. These are two different set of simulations. This one is high frequency storage data for shorter time scale, just to look at the water dynamics. Yeah.
That's why I was talking about hierarchy.
I see.
Maybe we can finish. We have to also get food. So,
Yeah.
So thank you. Suyash, what's your strategy briefly? So you can start.
Am I audible? Yeah. So I'm going to be talking about the effects of environmental exposures on health of humans as well as ecosystem. So let me just motivate the problem. So in 2005, C.P. Wild defined what was exposome. So it encompasses all the environmental exposure and individual phases or experiences during their lifetime. And this can be anything from environmental chemicals, dietary constituents, pharmaceutical stress, etc. Now we are going to focus on environmental chemicals, and let me tell you why. So the thing is, when you say environmental chemicals, then naturally you go to toxicology, and the current methods which are used in toxicology are limited for majorly for two different reasons. One is the ever expanding chemical space. So if one were to segregate the chemical space into measurable exposome space and identified chemical space, we see that the identified chemical space is a very tiny fraction of the entire exposome space. To put this in perspective, PubChem, which is one of the largest repository for chemical structures, currently maintains 115 million chemicals, and there are 350,000 chemicals in the market, of which only 10,000 have been characterized for their toxicological effects. And these mostly rely on animal testings, and you can see the number of animals being used in research is also very high. But these animal models also have some drawbacks, such as they poorly translate to human health due to interspecies species specific variations, and additionally they are time consuming and expensive. So in 2007, the US National Research Council came up with a landmark report titled Toxicity Testing in 21st Century, where they suggested we instead focus on toxicity pathways. Toxicity pathways instead of looking at the endpoints. And they also suggested we find alternatives to animal models in the form of in vitro and in silico methods. So in line with this particular suggestion, we have developed various different qualitative and quantitative tools in order to study these environmental exposures. So this talk is derived from two different studies. One is on petroleum hydrocarbons, which is relevant for ecotoxicology, and if time permits, I will talk about heavy metals, where we have looked at human health as well. So let me talk about the central framework here, which pertains to the qualitative part of the talk. So adverse outcome pathways have been endorsed by the OECD, and they capture toxicological mechanistic information starting with molecular initiating event where a stressor goes and interacts with some biological entity. Then we follow cascade of events across different levels of organization, cellular, tissue, organ, and at the end the outcome is some adverse effect. And these are linear pathways where each key event, which is each node, that is each node, as well as the links between them, have to be substantiated with some experimental or literature-based evidence. Now, you can see that if there are two different AOPs or adverse outcome pathways related to a single chemical, and if they share a node among them, we can construct a network out of it. And these networks are useful in order to identify which are the emergent and novel pathways which are not identified in these individual networks. And they also help in development of various different in vitro assays. These AOPs are stored in a repository called as AOP Wiki, and as of 2024 there are 501 AOPs within this particular repository. Let me talk about the chemical group of interest here. So the study was motivated by oil spills, and the major constituents of oil spills are petroleum hydrocarbons. So these are small molecules consisting mostly of carbon and hydrogen, and we curated 320 petroleum hydrocarbons from published reports. We then classified them as aliphatic and aromatic, and within the aromatic group we found that there are 15 US Environmental Protection Agency prioritized polycyclic aromatic hydrocarbons, and most of the results which I will be showing are for these 15 prioritized PAs. Now the question was, how do we link or how do we find out which of these AOPs are linked to which of these petroleum hydrocarbons? In order to do that, we relied on three different resources: US CPA's ToxCast, EcoTox database, as well as comparative toxicogenomics database. So these three databases provided us with endpoints associated with these chemicals, which we then linked with various different nodes or key events within these AOPs, and we were able to construct a network out of which. So this is the stressor and species network, stressor here being the chemicals, and we successfully found the association between 75 petroleum hydrocarbons with 177 ecotoxicologically relevant AOPs. These were then divided into various levels of relevance according to the evidence which was available. And in the highest level of relevance, we find that 10 petroleum hydrocarbons are linked to 33 adverse outcome pathways, out of which benzopyrene was the chemical linked to most of these adverse outcome pathways, and we considered benzopyrene for further study. So if we take all the AOPs associated with benzopyrene and look how they are connected, we see that they form a largest connected component consisting of 29 different AOPs. Now if you expand that and create a adverse outcome pathway network, it looks something like this. And this thing has around 160 to 170 nodes, and since this is a directed network, we can perform network analysis and calculate various different network based measures such as betweenness, centrality and eccentricity. So network analysis provides us with various critical events. For example, we found that the highest betweenness centrality was associated with apoptosis, which is programmed cell death, and highest eccentricity was associated with activation of AHR. Further, one can look at certain pathways, for example the pathway here highlighted in blue, and this pathway we studied with respect to fish, and we see here how population growth rate is reducing due to increase in DNA methyltransferase inhibition, and this particular pathway we were able to substantiate with evidence based on published literature. So that was the qualitative part. Now how do we quantify the risks associated with these petroleum hydrocarbons? In order to do that, we extracted data from EcoTox. Specifically, we extracted acute toxicity data, which is short-term data, as well as bioaccumulation data in the form of bioconcentration factor. And we constructed a chemical and species network, and we find that for acute toxicity case, 80 petroleum hydrocarbons linked to 221 species, and if we summarize this network, we see that most of these chemicals are affecting crustaceans and fishes, as documented by the database. Further, if you look at the bioaccumulation of these chemicals within these species, we see that polycyclic aromatic hydrocarbons were disproportionately accumulating in all of these species groups. The other thing one can do with this acute toxicity data is construct species sensitivity distribution. So these distributions take into consideration the sensitivity differences in species and are helpful in deriving something called as HC5 value, which is the chemical concentration which is hazardous to 5% of the species within the environment. We developed a workflow in order to curate this data, and we were able to curate data for 14 petroleum hydrocarbons in the acute case and 6 petroleum hydrocarbons in the chronic case. Now this is how a typical species sensitivity distribution looks like. This is for benzopyrene in the chronic case. Then we calculated the hazardous concentration 5% for 5% species across these chemicals in acute as well as chronic case, and this allowed us to rank these chemicals or prioritize these chemicals based on their toxicities. And we find that benzopyrene again shows up here as the topmost most toxic compound. Further, this data can be used to assess whether these petroleum hydrocarbons are posing risk to certain regions. So to do that, we curated data on 2 coastal water samples and 6 river water samples from literature in India, and we performed something called as risk quotient analysis, which tells us whether a particular region is at high risk or low risk due to the chemicals there. And these are the results. We see that for Mithi River, the risk quotients are very high, and then the question was, why should this be the case? And we went back and we looked at the literature to find that this particular river is highly polluted due to dumping of solid and industrial waste in it. So that kind of validated our analysis. So that was the quantitative part, and further what one can do is, as we have shown in another study, is to streamline this process, and we did that in case of heavy metals, and also expanded the scope of this study to human health. And I won't go into the details, but this is how the cadmium associated AOP network looked like, and we studied 4 different adverse outcomes, 2 specific to humans, which are decrease in lung function, increased kidney toxicity, and 2 in case of aquatic species, decrease in population growth and increased mortality. Further, the data from SSGs can be included in the toxicity stressor species network in order to give us more insight into which chemicals are which species are highly sensitive to which chemicals, and these links are highlighted in red here. Further, the results obtained from these studies, this study is available on this website, which is freely accessible. So to conclude, we developed network-based frameworks which are scalable in order to tackle the problem of the ever-expanding chemical space in 2 different ways: qualitative as well as quantitative. Qualitative based on AOP network, which tells us about the mechanisms of toxicity, and quantitative in the SSG or stressor species network based analysis, which tells us what are the risks associated with petroleum hydrocarbons for different species as well as different geographical locations. With that, I would like to thank Professor Samal, all my co-authors, our collaborators. So these 2 studies were done in collaboration with National Center for Coastal Research, MOES, which is in Chennai, and also the funding agencies IMAC and HPNI. Thank you.
So all this picture what you have shown, this you have drawn or
you mean this?
Picture?
No, we made this in Cytoscape.
Oh.
Yeah.
And it is a very if you have a size of data what you are handling, what is the size of data?
So if I talk about the databases here, so the data in this particular here we have
timing exactly.
The size.
yeah, so here we are handling.
Oh, thank you. I got got stuck in this DFS.
Size of data.
Yeah, so we for ToxCast we have around 10,000 chemicals. CTD has I think more than ToxCast, but I am not sure about the actual number here. EcoTox has around 13 to now it has around 14,000 chemicals and 13,000 species. And the chemical set which we had contained 320 chemicals, and this AOPs were around
But no,
320.
that is okay. After that you make this network.
Yes. So this is just a portion of the full network. Yes.
What you do next?
Okay.
You get network, this network, you
So once you
write
have
some
this,
code or something, what
yeah,
you
yeah.
do?
So once you have this network, you select a particular chemical. In this case, it was benzopyrene. You take all the AOPs associated with. So each of these nodes here is a pathway. Now, once you have that, so here we have all the AOPs associated with benzopyrene. Now each of these is a pathway, and there is a link between them if they share a node among them. Now you can expand these and you get this particular network, and this tells you about various mechanisms. So if you say activation of AHR, so here the chemical comes and binds to this aryl hydrocarbon receptor. So if that happens, what would be the downstream pathways that would be activated or downstream
This is
activities?
your numerical whatever code you have written, that is the output, right? This
Yes,
is your output.
so there are no dynamics here. This is just based on the topology of the
No,
network.
I'm not talking here.
Yeah.
Analysis
Previous
time.
Okay.
Some algorithm you have.
Yes.
That algorithm generated this, right?
Yeah.
Right? And then some software. You have
And after that, we calculate different measures. So these are the different network measures.
No, just I wanted what you have.
Okay.
Any other questions? Adijit seems very happy with this question. Okay, if there are no more questions, please thank the speaker again.
Thank the organizers.
Organizers, exactly. I mean, this, we have to thank two people here. Of course, who initiated all this.
I just want to say the quality of the presentations were really fantastic. Really. Please keep it up. I think you should assemble more often. If I may make a request. Because then you folks can share among yourselves. But the quality was